Imagine this for a moment. Every human being on Earth once existed as a single microscopic cell.
There were no eyes, no heart, no brain, no hands or feet. Yet within that tiny cell, the complete genetic blueprint of an entire human life had already been written.
The color of your eyes, many of your physical characteristics, and countless biological traits were encoded from the very beginning. But how does this extraordinary journey actually begin? Why does only one sperm succeed out of hundreds of millions? And what incredible biological events unfold immediately after fertilization? Today, we are diving deep into the fascinating world of embryology to understand how human life truly begins.
Human reproduction begins when hundreds of millions of sperm cells are released into the female reproductive tract during ejaculation.
A single ejaculation may contain approximately 200 to 300 million sperm cells, but only one has the opportunity to fertilize the mature egg.
Their journey is extremely challenging. The acidic environment of the vagina, the protective cervical mucus, eliminate the vast majority of sperm long before they reach their destination.
During this journey, sperm undergo an essential biological transformation known as capacitation. This process alters the sperm's outer membrane, making it capable of recognizing, binding to, and ultimately fertilizing the egg. Without capacitation, fertilization cannot occur, regardless of how healthy the sperm may be. Meanwhile, inside the ovary, a mature egg is released during a process called ovulation, which typically occurs once during each menstrual cycle. The egg remains viable for only about 12 to 24 hours after ovulation. If a capacitated sperm reaches the egg within this narrow time window, fertilization becomes possible.
The egg itself is protected by multiple layers. The outermost layer of cells is called the corona radiata, while beneath it lies a thick glycoprotein shell known as the zona pellucida.
These protective layers prevent unwanted cells from entering and ensure that only a properly prepared sperm can reach the egg.
When the healthiest sperm finally reaches the egg, a remarkable event begins. The sperm carries a specialized cap on its head called the acrosome, which contains powerful enzymes.
These enzymes gradually digest a pathway through the zona pellucida. This allows the sperm to approach the egg's plasma membrane.
As soon as the sperm successfully fuses with the egg immediately triggers a protective mechanism called the cortical reaction. During this reaction, tiny cortical granules beneath the egg membrane release their contents, causing the zona pellucida to harden almost instantly. This prevents any additional sperm from entering the egg, ensuring that only one sperm contributes its genetic material. This phenomenon is known as the block to polyspermy, and it is essential for normal embryonic development. Inside the egg, the nucleus of the sperm and the nucleus of the egg gradually move toward one another. Each contains 23 chromosomes. When these two pronuclei fuse, they restore the normal human chromosome number of 46, creating the very first cell of a new individual, the zygote.
At this precise moment, a completely unique genetic identity is established. The biological sex of the embryo, along with thousands of inherited genetic characteristics, has already been determined. Although this new life consists of only a single cell, it possesses all the genetic information required to build every tissue, every organ, and every system of the human body. This single microscopic cell marks the beginning of one of the most extraordinary biological journeys in nature. A journey that will transform one cell into trillions of highly specialized cells, ultimately forming a complete human being. Although the zygote has now been formed, the journey of human development has only just begun. This single cell immediately starts dividing through a process known as cleavage. Unlike normal cell division, cleavage rapidly increases the number of cells without increasing the overall size of the embryo because it remains enclosed within the protective zona pellucida. About 24 to 30 hours after fertilization, the zygote divides into 2 cells. Soon afterward, it progresses to the 4-cell, 8-cell, and eventually the 16-cell stage.
At approximately the 8-8 cell stage, the embryo undergoes an important event called compaction,
during which the blastomeres become tightly packed together. This is the first step towards cellular specialization, allowing the embryo to distinguish between inner and outer cell populations that will later develop into different tissues.